Hey everyone, and welcome to our third lecture from the International Youth Neurosciences Association's Introduction to Neuroscience summer course. Over the past two lectures, we learned about the basics of the structure of a neuron and how signaling moves within a neuron in our action potential lecture. And today we're going to have the pleasure of hearing from Professor Yuong Li, a professor of life sciences at Peking University, on how neurons communicate with one another.
Okay, let me just get started. All right. So first, a brief introduction about myself. I'm currently at Beijing in Peking University. I originally got my bachelor degree here. And then I went to the US at Duke University. I got my PhD degree in Department of Neurobiology, and then for my postdoctoral training at Stanford University. And I came back here more than 7 years ago to set up my own research group. And my research interest is about how neuronal communicates. And specifically, we actually focus on studying neurotransmission, but we also developing tools. We are also developing tools to visualize how this neuronal communication might occur, and using that in the in vivo systems to sort of probe or sort of spy on the neuronal communication in real time, and that can be very helpful to understand what happened in a physiological condition and what might go wrong in a disease condition. And I also gonna sort of briefly talk about, in the previous two lectures, different speakers covered the sort of cell of the nervous system and the mechanism of action potentials. And today, my main topic, and I understand most of you are high school students, are gonna talk about what's the mechanism of neural communication, neurotransmission, and that is how neurons, when they sort of generate action potential, which can propagate to the sort of the end or the axonal terminals, and that will trigger the transmitter release. And what sort of mediate this process, and specifically the calcium influx that due to the generation of sort of action potential and the depolarization sort of causing by the action potential will open up the voltage-gated calcium channel. And then once the transmitter being released, and what kind of transmitter might sort of exist in the nervous system, and how they get sort of released. And in fact, they are mostly loaded in the synaptic vesicles and then being released through a very sophisticated regulation. And once they release, they will activate the postsynaptic receptors, and what kind of receptor it is. And they are excitatory, inhibitory, and also there are different time course for this receptor sort of action. And the release transmitters can also be sort of passively diffused out, but also being actively degraded in a way to ensure the sort of fidelity of this transmission. And also, a lot of my PowerPoints actually are coming from this very good book, Principles of Neurobiology. And this is the first edition, and again, authored by a single author, in fact, Dr. Li Qunluo. And it's a not so big book. So for this complex sort of a system like a nervous system, this is actually good because it could be like 3,000 pages, a big book, but it's probably will be very challenging for high school students. And also they have he has the second edition coming, I believe, with the color green in this August. So, okay, so neuronal communication actually occurs through the so-called synapse, but there are two sort of different kinds of synapse. One is the chemical synapse, which we are gonna mainly be discussing about, but there's another type, the so-called electrical synapse, where the cells that's on the right in blue, where the cells that they connect with each other through these gap junctional channels. And these physical gap junctional channels will allow currents. For example, the action potential generate currents will be rapidly sort of coupled with each other. And this will allow really fast almost instantaneous sort of electrical coupling. And this is important, for example, in our heart, where the heart cells, between different heart cells, they also have gap junctions, and the contraction that will be synchronized by this sort of gap junctional coupling to enable that the heart cells to pump blood. In the nervous system, there's also some neurons, they have the gap junctions, and that will ensure them to have this synchronous firing. So that would be the gap junction. But majority of the neuronal communication in the nervous system actually is through the chemical synapse on the left, which we are going to be sort of specifically discuss. And if you look at this chemical synapse transmission, again, through this more than 50 years of intensive research, we already understand quite well the mechanism of this synapse transmission. That is, if you look at one here, the action potential that's sort of been generated, and again, you guys already learned that, and that's the interplay of voltage-gated sodium channel and voltage-gated potassium channel in this special way to generate this all-non action potential that will propagate to the nerve endings. And at the nerve endings, this action potential will depolarize the plasma membrane, and there resides voltage-gated calcium channels, which is sensitive to this depolarization. And once it senses this depolarization, that it will open up, and it's very selective and allows the calcium influx to get into the cell. And this localized calcium influx will trigger the transmitter release from the so-called synaptic vesicle, and that's this teeny tiny vesicles with a diameter about 40 to 50 nanometers and loaded with very rich and high concentration of transmitters. And this calcium triggering process that we are going to discuss in detail a little bit later will cause this synaptic vesicle to fuse with the plasma membrane, and the transmitters will be discharged from the vesicle and diffuse through this synaptic cleft here. And this synaptic cleft is teeny tiny, nanometer in range. And even though it's diffusion, but with such a short distance, so it actually sort of takes a very short period of time. And it travels through this synaptic cleft, will bind to the cognate postsynaptic receptors, either ion channel or GPCR, as we'll mention later. And this postsynaptic sort of receptors will be activated or will open up, and depends on the receptors. If they are ion channel, they will allow either sort of a positive ions get into the cell, and that is so excitatory sort of postsynaptic transmission, and that will, because the positive ion will go into the postsynaptic cell, it will cause the postsynaptic membrane. Originally, so there is a negative inside, but you have the positive ion, it will depolarize. That is, the membrane potential will become less negative, okay, and that will cause the cell to generate action potential once the membrane potential reach the threshold for the postsynaptic cells, and that will further propagate through this cell. But the transmitters will also be inactivated by different pathways, depends on transmitters, and they can either sort of be reuptake by some transporter expressed in these neurons or the adjacent glial cells, or there's some specialized enzymes that are sort of evolved and just located near the synaptic cleft and rapidly degrade this transmitter. For example, acetylcholine, it has a sort of specialized esterase which just sort of cleave this acetylcholine and then degrade it, and therefore this transmitter is sort of no longer, once been cleaved, no longer able to activate the postsynaptic receptors. And the specialized of synapse, for example, the majority of the synapse in our brain is the neuronal communications, but also neurons can innervate muscle, and this special synapse called the neuromuscular junction and has these unique structures. Okay, for example, if you look at the motor neuron, that is sort of their cell body sort of in the ganglia near the spinal cord, but it will send its axon to innervate the muscle. And the muscle, you can see that originally the axon will be sort of covered up by the myelin sheets, but at the sort of the connection sites, the so-called neuromuscular junction or the end plate. Okay, the end plate region that it doesn't have this myelin sheet. And if you want, you can look at the EM, and again, this is one of the sort of drawing. You can really see these teeny tiny vesicles that containing the acetylcholine, and there's this postsynaptic acetylcholine to the ion channels. So these imaginative-like sort of structures that enrich a lot of acetylcholine sort of receptors, and you will receive the discharge of acetylcholine and activate the muscle cell, and again, if it reaches the threshold, will cause the muscle cell to sort of contract, and that's the sort of neuromuscular junction. And in fact, a lot of our understanding of this synaptic transmission are initially really derived from the experiments in the neuromuscular junction, for one of the reasons, which is that the muscle cells are relatively big, okay, and therefore it's relatively robust. If you poke electrode to record sort of the electrical signals that sort of in the that elicitate in this muscle cell, is relatively robust and easy. Okay, and in neurons, and it takes a lot of techniques in the central nervous system. Takes a lot of technique development. For example, people getting Nobel Prize by inventing patch clamp that allow one to record these teeny tiny neurons with their, for example, cell body about like 10 to 20 micron. And it turns out a lot of the principles derived from the muscle cells actually are conserved in the neuronal communications, and therefore sort of this also illustrates how one can use a sort of a model system to have a very deep understandings initially at least, and then apply them generally across different preparation, and then to derive the common principles, and also save one's efforts to have some initial breakthroughs. And again, as I mentioned, that the action potential that sort of arrive at the nerve terminal, and a lot of studies are really studied from neuromuscular junction. For example, this is the sort of class derived from the classic work from people done in 1950s, studied the neuromuscular junction where the acetylcholine, the structure shown in A here, that is the transmitter. And one of the experiments that demonstrate that acetylcholine indeed is the transmitter at this synapse is through the following experiment in B. Well, if you use the sort of the stimulation electrode showing here on the top, stimulate the motor axon. And if you recall from the intracellular electrode shown in green, you can observe once you stimulate, there's a sort of membrane potential change that you can record. Okay, and that's how you activate this neuron and the transmitter be released. And in fact, if you simultaneously pose a sort of a patch pipette filled with acetylcholine inside the pipette, and then you use this sort of so-called electrophoresis to inject or puffing the acetylcholine near this end plate, you can get this almost identical response. Okay, demonstrating that by puffing this acetylcholine sort of rapidly, you can mimic the effect of stimulated. So this is one of the key sort of experiments demonstrate that the acetylcholine is the transmitter for this motor axon that sort of innervate muscle in the mammalian system. Okay, of course, this is not the only evidence, and this evidence alone actually is not sufficient. Okay, for example, if we use some other artificial sort of ligand that people generate that can activate nicotinic receptors, and then if you do that in the same way to path into the end plate, you can also activate the nicotinic receptors, and you can also generate a similar effect. For example, the post-decompensation. And because that substance is artificially generated, doesn't exist in the sort of nervous system, so it doesn't really to be the endogenous transmitters. Therefore, there are other evidence that people can detect acetylcholine in the nerve terminals, and also if you remove or eliminate acetylcholine in the nerve terminal, and if you stimulate the neuron, you don't see that effect. So, coupled with those sort of loss of function experiments, and one can really reach the conclusion that it's the acetylcholine that is the transmitter that, in this case, that sort of mediate the neurotransmitter release in the neuromuscular junction. And what is the fundamental unit of the transmitter release? And when Bernard Cass did experiments in 1950s, again, Bernard Cass got a Nobel Prize by sort of help us deeper understanding of the mechanism of transmitter release. And this is one of the experiments they did. So if they carefully record, again, from an end plate, and then also in a way to reduce the transmitter release, they found actually they can observe this teeny tiny bump here. And this teeny tiny bump, sort of, if they quantify them, they have a relative sort of homogenized sort of homogenized amplitude. Okay, for example, if you sort of treat them with Gaussian curve, you see that they all sort of peak at the center 1 here, and of course, from time to time, you also see there's a second peak, and through careful mathematical measurement and also some experiments, they found that the postsynaptic, this is small membrane potential, they call the miniature end plate potential, MEPP, miniature end plate potential, that is sort of quantized. Okay, and that is with the equal size, and the second peak and the third peak can be explained that they are simultaneously, that there are two of those units that overlap with each other, and then you can see these two peaks. Okay, and one might wonder maybe that as this small unit-sized peak is corresponding to a single acetylcholine release. It turns out it's not. Through careful measurements that they, through calibrations, for example, Dr. Koffler did, that they actually estimate that more than 1,000 of this acetylcholine in that small packet. And through more complicated experiments that I don't want to go into detail, they conclude that this is the quantal sort of a transmitter release of acetylcholine. And almost at the same time, the development of the electron micrograph, okay, that is using the electron to look at the microscope and to look at the neuromuscular junction preparations. People readily observe at this synapse. For example, this is the neuromuscular junction presynaptic axon, and this is the postsynaptic sort of a muscle, and you can see this invagination, and this is the section of that. You can observe there's these teeny tiny vesicles at this axon, okay, and there's a special size that opposing the postsynaptic muscles, and this single vesicle corresponding to the MEPP sort of a potential they observe. and there is a single vesicle being released that will discharge its content and activate the postsynaptic receptors. And in fact, for example, our nervous systems, and the synapse is more like, looks like on the right, okay? And that is the neuronal synapse. And for example, on C here, you can see that is the presynaptic axon. Again, there is the synaptic vesicles with the similar shape. And also there's this dark electron-dense regions that sort of connecting or opposing to the postsynaptic neuron. And one can observe both the postsynaptic side here, there's electron-dense sort of material marked by stars. But one can also sort of seeing that there's also electron-dense material at the presynaptic membrane. And that is the typical excitatory sort of synapse. That is the presynaptic neuron containing glutamate in their vesicles, sort of opposing the postsynaptic neuron with postsynaptic density, and there is a spine. Okay, and again from this one section, and you probably will be asked, how do you know it's a spine? Okay, and again, people can do this serious section and then reconstruction in the 3D, and one can see again. I will talk about later. Is a protrusion of the sort of spine that from the So neurotransmitter release requires the transmitter sort of packed in the vesicles, and this single vesicle corresponding to the fundamental unit. And every single time the neuron can release one or not no vesicle or more vesicle, they cannot release the transmitter less than a vesicle. Okay, so that's the so-called quantum synaptic transmission. And then what are the materials inside of the vesicle? Again, recent work, and this is a work from 2006, people purify those vesicles and then using the sort of cutting edge, for example, mass spectrum analysis, and they can really look at what materials are in or located in the vesicles. And for example, and this is the cartoon illustration of how the vesicle and their protein distribution. Okay, and one of the important things here, the VATPase. The V means vesicular ATPase. The ATPase means that you will hydrolyze ATP. Again, ATP is the energy currency inside the cell. And this VATPase that will use ATP as a currency and hydrolyze, and the hydrolyzation will make this protein move, and it will pump proton inside the synaptic vesicle. And therefore, almost all the synaptic vesicles are acidic. They have high concentration of proton inside. pH is lower, like 5, 6. Okay, it's much lower than the neutral pH 7. Okay, there are proton inside, and this proton is the proton has a high concentration inside because of the vesicular ATPase. And this proton, as well as the charge that generated by the proton has a positive charge, okay, and it has a chemical concentration which is higher inside. So the chemical energy and the electrical energy of the proton will also be used by the vesicular neurotransmitter transporter. So this transporter, for example, they can use the proton's energy. Okay, it has high concentration, and it has a positive charge inside, and that can be used directly or indirectly by this transporter to load the transmitter. For example, acetylcholine, for example, glutamate, depends on different neurons, and to enrich the concentration of the transmitter inside the vesicle. Okay, so the concentration of the transmitter inside the vesicle are very high through this energy-dependent process. And there's also additional protein, for example, synaptotagmin, that we will touch upon. They are the important protein that can mediate the vesicular to plasma membrane fusion, okay, by pairing with, for example, syntaxin and SNAP25, the so-called SNARE protein in the plasma membrane, and this energy will drive the membrane fusion. Okay, there's also Synaptotagmin here, and again I will talk about a little bit later. Those are the sort of calcium sensor, and that will be sensing the calcium influx that I mentioned a little bit earlier, and then through interaction with snares and the plasma membrane or a vesicular membrane lipid, and it will cause the membrane fusion. And there are also RAP3 and additional proteins. There's still some of those proteins that we don't have a clear understanding of their function. Even note that we know they are there, but we still have no idea about their function. And again, that will be still under intense studies, and understanding that can help us to understanding the process. And as I will also mention later, by understanding this process in detail, we can design therapeutic approaches, for example, in some disease condition, and to repair the system. And how this transmitter, the synaptic vesicle, get fused? Okay, I mentioned that the synaptobrivin in a previous cartoon. In fact, the synaptic vesicle has this blue synaptobrivin protein, and it will pair with this sort of red called the syntaxin protein. I'm not sure whether you can see on the right here. Okay. Let me just make it smaller. Okay, there's a red syntaxin here. There's also sort of green SNAP25 here, and it forms these 4 helical bundles. Okay, and through the triggering by Synaptotagmin, calcium triggering, the SNARE complex will assemble, and it's thought to be like this zippering sort of process. There's the zip, it's like a zipper that will drive the sort of the synaptic vesicle membrane fuse with the plasma membrane. And once it fuse, because you have a high concentration of the transmitter inside the vesicle, and the diffusion will drive the transmitter to diffuse through the fusion pore, and then through the synaptic cleft and activate the postsynaptic neuron. And again, one can talk for example, a whole semester about the detailed pathway, how people identify. How do you know it's the sort of synaptobrivin? And the corresponding part is the syntaxin and SNAP25. Again, that's a really interesting discoveries that people make, and people getting Nobel Prize for that. For example, Gene Rossman and Tom Schodorf, they are getting Nobel Prize by help us understanding at molecular terms what are the essential protein and what is the process and how it's being regulated. And as on Brugger here, this here, using the sort of structure study and then crystallize this complex, and therefore we can see at atomic resolution how these 4 helical bundles assemble, and which amino acid and which atom sort of interact with the other atoms. Okay. And how do we know indeed is the calcium trigger the transmitter release? Well, again, one of the experiments, let me just show you here. There are many evidence study from 1950s or earlier. And this is one of the experiments, probably is the sort of the first using the fluorescence to imaging calcium and then demonstrate that there is a calcium really at the presynaptic terminal that will sort of get influx during the nerve activity, and that can trigger transmitter release. And showing here is a special synapse from squid. And it turns out squid has this unique giant synapse that are very, very big, and you can look at the scale here. That this is is 500 micron, 0.5 millimeter, and in fact the squid here is the why I was illustrated at the postsynaptic membrane, and this is the postsynaptic axon for this screen to mediate its escape behavior. If we facing danger, they will just sort of contract all the muscle, and this is the neuron that is sending out the signal, and it will squeeze water and go backwards. And this is the giant postsynaptic axon. And in this axonal synapse, this presynaptic axon will innervate multiple postsynaptic axons. But if we just look at one here, and if we use the calcium dye that we pre-inject into the axon, and the dye will just emit photons once the calcium level is high. Okay, and you can observe that during you stimulate this presynaptic axon, and that's really high concentration of calcium that sort of especially facing the postsynaptic cell here, and there's a really high concentration of calcium at this side, at the synaptic side. And even though the whole presynaptic axon is activated, but the calcium level again is concentrated at the side near this postsynaptic neuron. Again, right now we actually know there's specialized calcium channel through really sophisticated regulation anchor at the presynaptic terminal to really allow the calcium influx to be very close at those synaptic vesicles, and where they have the calcium sensor synaptotagmin, and then will mediate this rapid membrane fusion. And also, if we look at in time through the experiments, okay, and again, this is a little bit complicated for high school students, but one can use methods to really control the calcium into the cell, okay? And on the top row showing that, for example, if you use a sort of so-called voltage clamp to control the membrane potential of the presynaptic neuron, and you can sort of jump up to the membrane potential through voltage clamp from initial resting condition to depolarize a little bit to the, for example, to minus 25 millivol. And this sort of depolarization or less negative membrane potential change will open up the voltage-gated calcium channel. And sure enough, if you use the electrophysiology record, indeed you can see that's this calcium current. Again, calcium has charges, and if charge ions going through the membrane, and then you create currents that can be captured by your recordings, and you can see there's a calcium influx. And with this calcium influx, and if you simultaneously record at the postsynaptic membrane, you can see there's a transmitter being released and activate the postsynaptic membrane, and the postsynaptic cells, there's an ion channel be opening up, and therefore you can record a excitatory postsynaptic current. That is, if you artificially sort of change the membrane potential, depolarize a little bit, it will activate the voltage-gated calcium channel. You can really verify, and you can record by your calcium current measurements. And then with a short delay, again, the time scale here is 5 milliseconds, so it's indeed with like 1 millisecond delay, you can immediately observe there's a postsynaptic neuron being activated. And this delay actually includes the sort of the time for this calcium comes in and then binds to the calcium sensor, and the calcium sensor will trigger, and then trigger the membrane fusion, and then the transmitter need to be diffused, and the diffused transmitter need to bind to the postsynaptic receptor, and postsynaptic receptor also need to open, and then we can do the recording. And therefore, for our survival, or for animal to survive, we really evolve very sophisticated protein machineries that regulate this process to make it really rapid, okay? And for example, a predator and prey interaction, if you are no response is rapid enough, you are just going to be die and select throughout the evolution. And one can also argue maybe it's just the voltage, it's not the calcium channel that trigger the opening of the voltage-gated calcium channel that trigger the transmitter release. This is not the case. And if one, in a sense, sort of preparation to the experiment, you no longer depolarize to the minus 25, but you depolarize into a more positive membrane potential. You are still sort of changing the membrane potential. And for example, in the plus 50 millivolt, and in this case, there's no longer transmitter being released during this depolarization. You don't see that, okay? And there's also no calcium current. So there's no calcium current and there's no transmitter being released, even though you are depolarizing the membrane, okay? And some of you probably might ask, why in this case there's no calcium current you can detect? Well, if you still remember some of the sort of knowledge you study in the action potential, the ions going through the membrane really depends on its electrochemical gradients. If we are sort of deprive the membrane potential to really, really high, even though calcium has the chemical gradient wants to go inside once the calcium channel opens, and calcium channel indeed open, but because you have a lot of positive charge by depriving the membrane potential, and it will counter the calcium influx. So in fact, the plus 50 millivolt is close to the so-called equilibrium potential of calcium ions. So there's no calcium net influx. Okay, so in this case, this set of experiment demonstrated it's not a depolarization of membrane, but it's the calcium that get into the cell can trigger. And at the end of the experiment, if you go back the membrane potential to the resting membrane potential, and again, the calcium channel still open and then gradually close. But once the membrane potential goes to the -70s, now the calcium ions do not have this positive charge to prevent from coming in, and it will just rush in, and therefore you can record this so-called tail current of calcium, and that can also trigger the transmitter release. Okay, so sort of these experiments that one can really look at in a quantitative terms how calcium influx that can trigger transmitter release, and it's the calcium and not the membrane depolarization per se that trigger the transmitter release. And so what is the molecule? And I mentioned through, for example, Tom Schulter's work, they demonstrate it's a synaptotagmin. How can they demonstrate that? Well, one of the evidence comes from their knockout experiment. If they, in the mice, and knocking out just the synaptotagmin, okay, using the gene knockout techniques, which was cutting edge in the early 1990s. And if you record the wild-type neuron and record the sort of control neuron, the wild-type neuron and the knockout neuron, and look at the difference, and you can see that in the wild-type neuron, if you stimulate the presynaptic neuron, you can see this really sort of synchronous transmitter release by electrophysiology recording by the Synaptotagmin 1 knockout. This sort of a large synchronous calcium trigger release component is gone. Okay, so this data is consistent with Synaptotagmin 1. This is the calcium sensor that sort of mediate the transmitter release and the calcium sensing part. But it doesn't prove. For example, if the one of the synaptobrevin, the SNARE protein, that really mediate the fusion, if you do that knockout, you also don't see the transmitter release. And for example, if there's some special gene that is important for generation of those teeny tiny vesicles, if you are knocking that out, and if you do that experiment, you also might observe there's no transmitter release. Okay, so they do much more further experiments. And one of the key experiments actually was done also relatively recently. For example, that is already close to 20 years ago. They can introduce a single point mutation at the synaptotagmin. And in this point mutation that is close to the calcium binding site of synaptotagmin, and then you can measure the calcium binding ability in the test tube, and you found that the calcium binding ability that sort of shift to the right, that's less calcium sensitive, for example, by 2-fold. And then if you introduce this point mutation synaptotagmin and replace all the wild type inside the sort of in the mice, and then you do the experiment to look at the calcium mediate transmitter release assay, and you found just corresponding to your biochemical alteration of the synaptotagmin binding to calcium ability, the physiological elite state transmitter release, the calcium dependency also shifts proportionally to the right by the same amount. So this sort of experiment more convincingly demonstrates that the synaptotagmin indeed is the calcium sensor. That is, if you alter the calcium binding ability just by the amount, like twofold, and in the in vivo neurons, it also changes the calcium binding of affinity without changing other properties. Okay, and indeed, the sort of the current model for the transmitter release, people getting Nobel Prize for that, is we already have a lot of deep understanding at the molecular terms. There is some specialized SNARE protein, synaptotagmin, located at the synaptic vesicle. It will pair with the SNAP25 and syntaxin again. Sorry for the complicated names. And that will sort of mediate the membrane fusion, but before the calcium triggering, they are just ready right there without zippering. And synaptotagmin will, through this calcium binding site, the so-called C2A and C2B domain, okay? And again, they bind more than one calcium actually. And that will interact with the SNARE complex as well as the lipid membrane will cause the synaptic vesicle fusion. And this system is designed to react really rapidly to mediate the physiological release. And let me switch gear a little bit. What are the transmitters? I mentioned acetylcholine. That is one of the most well-studied ones because that is the first sort of transmitter be identified more than 100 years ago by Dale and others. Okay, but there are additional transmitters in our brain. The majority of the so-called excitatory transmitter are glutamate. That's in our brain. In our muscle, the motor neuron is using the acetylcholine. Okay, and GABA is a transmitter again derived from amino acid. Okay, and without with some small modification, the GABA is the major so-called inhibitory transmitters. Okay, and in addition to these two, there's other major excitatory and major inhibitory in our brain. There's also additional transmitters. For example, glycine is also inhibitory, but mainly in the spinal cord or the brainstem, not in the sort of cerebral cortex. And there's also sort of dopamine, for example, important for our movement and important for addiction and reward learning. Norepinephrine, which is again structurally similar to dopamine with only one hydroxy difference, and that is also important for the stress response. Serotonin, famous for, it's a drug target in the depression. Okay, and actually the serotonin transporter is a major drug target that can sort of reuptake and serotonin. There's also histamine and others. And here listed the common transmitters and there are sort of different locations. And again, the mammalian system and the invertebrate system are sort of different. At least the location are different. Okay. In addition to these small transmitters that are on the top, at the bottom here, there's also neuropeptides. Okay, and the neuropeptides are sort of another larger family of neurotransmitter and neuromodulators. And for example, they are believed to release relatively slower, but very important for different aspects of physiology, for example, hunger, aggression, and we will talk about it a little bit later. They will work by targeting mainly the G protein coupled receptor, these 7 transmembrane proteins, rather than the ion channel itself. Okay, and in addition to this sort of membrane fusion process I just mentioned, for example, I mentioned the blue ones are the synaptotagmin, and there are syntaxin and SNAP25 here. There's additional protein that's in the synaptic vesicle, and there are some of them we have some understanding of their function. For example, MONK18 help to regulate the snares. Synaptotagmin will bind to calcium and then interact with the snares, the calcium sensor. There's also this small GTPase called REP3 that can direct or indirect sort of interact with this large protein called sort of scaffolding, presynaptic scaffolding protein called ring or ring binding protein. This large protein can also further interact with, for example, voltage-gated calcium channel, and therefore through this sort of cross-linking or sort of cross-interaction, it actually can link the calcium channel and skeletal vesicle relatively close in space. And therefore, once the calcium comes in through the voltage-gated calcium channel, in its close proximity, and there is the vesicle and the synaptotagmin right there to trigger the transmitter release, and you can make it very efficient. Okay, and there are also sort of a presynaptic adhesion molecule that can interact with the postsynaptic adhesion molecule to make this tight synaptic cleft. As I mentioned, there's also only nanometer sort of distance between these sort of two adjacent presynaptic and postsynaptic neurons. And also there's a recognition problem that people might wonder, right? That different neuron, different synapses, and how the neurons can wire in a logical way to have this coordinated function. And in fact, this sort of neural adhesion molecule, I believe, play an instrumental role, not only mediate this sort of close interaction, but also the different sort of isoform or different flavors of them that can mediate this recognition and to enable the right one will be targeted to the right postsynaptic neurons to have this correct neuronal assembly during development. And if there's something wrong with this recognition, people might have some disease. For example, there's some autism-related disease, autism spectrum disease. People found there's something wrong with this sort of neural adhesion molecule, and through the development, there might be some sort of misassembly of those synapses. Again, this is a field still under intensive study, and one can also use electron micrograph, and most recently actually can use this sort of so-called super resolution to look at this sort of a well coherent organization of this synapse. And through that, for example, one can look at the synapse, and this is a cartoon showing how this synapse might look like. The postsynaptically, there's this glutamate receptor that will sense the transmitter, but at the presynaptic at vesicles that can link with the propyloid, which is a scaffolding protein corresponding to some of the similar protein like a ring protein and other protein. The propyloid is the protein in the Drosophila, the fruit flies, and it has a similar architecture synapse like the mammalian synapse. And also this ring binding protein will further sort of link, serving as a bridge, link the calcium channel to this scaffolding protein and scaffolding protein touching the synaptic vesicles. And once the transmitter being released, again, I mentioned earlier, is really use the energy from this the ATP to generate the protein to pump the transmitter inside the vesicle. Once the transmitter being released, it binds to the receptor, but there's also sort of a sophisticated mechanism evolved through the evolution to remove them. Why do you want to remove it? Well, if you think about, for example, if you want to move your hands and your motor neurons need to control your muscle, not only you need to sort of reach, you need to also withdraw. So the muscle need to be rapidly controlled to contract and contract next time. And if you always have the transmitter once released hanging around, it actually prevent the next action. Okay, and therefore the nervous system actually use the different mechanism. For example, for glutamate, and there's this transporter, the PMT label here. That's the plasma membrane transporter that uses, again, chemical energy, in this case, sodium, in a way to react.
Exercise, correct posture, and
Hello?
race are
Hello? Somehow I have some noise here. Okay. So where am I? Okay. There's this transporter uses this chemical energy to sort of reuptake against the transmitter's chemical energy. Okay, for example, in this case, using sodium to reuptake the transmitter, like glutamate, like serotonin, like dopamine. They will be transported by the plasma membrane transporter inside the cell, and the cell can also reuse these transmitters. Okay, but also there's acetylcholine I mentioned that actually was not immediately recycled, but was rapidly degraded by the choline S3 enzymes to cleave it. And now, so that is sort of a pretty much of it about the presynaptic part of the story. Let me just sort of a summary a little bit here. Previously, what I was just mentioned was all the presynaptic mechanism. Okay, that is how the transmitter being sort of packed or uptake or transport packed with the transmitters, the vesicles, and the vesicle uses the very cool molecular machinery to sort of mediate the fusion, and the fusion process also involved this highly coordinated sort of synaptic structure where there's voltage-gated calcium channels, and there's this scaffolding proteins, and also how this calcium can be sensed by synaptotagmin to trigger release and transmit a different kind and then being released and being degraded. But how this release transmitter at the synaptic cleft diffuse and activate the postsynaptic receptor? Now let's just focus on this part. Again, one of the most early studies and well understood studies is from the neuromuscular junction, because of its easy accessible, and also because of the acetylcholine is the first identified transmitter. So people just have the ability to study them for more long period of time. So how this released acetylcholine lead to the postsynaptic membrane potential changes? This illustrates the experiments that the people done, again, starting from 1970s. And in this experiment, to more control the transmitter release, people can inject ionophoresis, the acetylcholine. And acetylcholine, if you look at its chemical structures, is positively charged. So if you use the sort of ionophoresis to inject them, it's actually relatively easy. If you just put the positive charge inside your sort of injection pipette, and that acetylcholine will just be sort of puffing out. Okay, and you can have very high concentration of acetylcholine, and therefore even you can puffing out a little bit, it doesn't change it of the net concentration inside the pipette, and therefore every single time you can puff the same amount of acetylcholine. And if you do that experiment, every single time you just puff the defined amount of acetylcholine. But if you change the postsynaptic, in this case, the muscle cells membrane potential, again through this technique called voltage clamp, and you measure the muscle response, and you find interestingly in figure B, that's what you found. Okay, if you change the so-called clamp membrane potential, and you found that if you clamp the membrane potential at 0 millivolts, you don't see any response. Okay, and basically you do this curve and plot in figure C here. What you observe, there's this close to a linear curve going through the 0. Okay, close to. Okay, so that is if you are really changing the postsynaptic membrane potential, you actually can observe the acetylcholine can elicit different current and voltage relationship. And the curve going through zero, as I mentioned earlier. And this zero membrane potential, again, if you remember the study about the action potential in the previous lecture, this is the membrane current zero. This is the so-called equilibrium potential for this acetylcholine receptor in this muscle cell. Okay, it's called equilibrium potential, or it's called a reverse potential. That is acetylcholine, if you put it, and the postsynaptic acetylcholine receptor being activated, the acetylcholine receptor is ion channel, and you will allow ion to go in through it. But the property of this ion channel is that you will allow this ion to go through with the reverse potential at 0 millivolt. Put it in a more understandable way, people actually can use this reverse potential to estimate what ions going through this ion channel, going through this acetylcholine-gated ion channel. It actually is the positive charge ions, the cations. Okay, because the cations, both the sodium, potassium, calcium can go in through it. Because these ions together has the property that it will reverse at 0 millivolt. Okay, and also this property tell us that how this acetylcholine can really sort of activate the receptor to work on the postsynaptic neurons or muscles, because once this acetylcholine receptor opens, you will allow calcium, the positive ion, to go through it, and the positive ion's reverse potential, or the equilibrium potential, is 0 millivolt, and all the neurons before stimulation is at the resting membrane potential minus 70 millivolt, and now you have the 0 millivolt if the acetylcholine receptor is being activated, and this activation will lead to the membrane potential to be depolarized. Okay, from minus 70 close to 0, right? It will depolarize, and once it goes into the depolarization, if it reaches the threshold, it will generate the action potential. Okay, so the acetylcholine is working as an excitatory transmitters because it makes the postsynaptic membrane potential more close to the threshold to cause the postsynaptic neuron to fire action potential. So that is the mechanism of how this acetylcholine really work by activating the nicotinic or this ion channel. Okay, as I will mention a little bit later, that acetylcholine, just like a glutamate, it can activate different kind of receptor. The ion channel itself is called nicotinic receptor. It's defined by its agonist. That is, people found there's some artificial sort of chemical nicotine, okay, that you put it in the, for example, neuromuscular junction preparation. You can mimic the acetylcholine effect. Okay, so it's the nicotine-like ion channel, and this is really illustrate why I just mentioned again. Okay, the acetylcholine can activate the postsynaptic acetylcholine receptors, and the acetylcholine receptor, through the Bernard Katz measurement, okay, he's a pioneer, that it will allow the positive ion, and in this case, mainly sodium, has a high concentration, and that will lead to the depolarization of the postsynaptic membrane, and it will have the sort of the excitatory effect. And again, through a lot of research, people first notice this effect. Okay, and through a lot of research, nowadays we actually have molecularly clone and identify what is the molecular nature of those acetylcholine receptors. For example, people have already cloned the acetylcholine receptor and found out that it requires 5 subunits to assemble into an ion channel. Okay, and the 5 subunit is also called pentamers. Okay, and through this sophisticated assembly, that you will allow it will form a pore, and the pore will be regulated by acetylcholine, and acetylcholine once binds to it, it will open up this pore and allow sodium and other positive ion to go in through this pore and depolarize it. And in fact, by understanding the molecular natures of acetylcholine, we have a better understanding of how some toxin might work. For example, here on B is showing the experiments if you using the ionophoresis to puff acetylcholine. And you found that if you pre-treat the muscle with curare, this acetylcholine elicit nicotinic ion channels current is gone. So put it another way, curare actually is the toxin that will prevent acetylcholine ion channel from opening. Of course, some of you probably don't know, but curare is actually a toxin that is being identified from plants and being used by South American or Central Americans more than 100 years ago. They put these ions, this toxin in the arrows, and then using these arrows to sort of in a way to for hunting. Again, at that time they have no idea what is the sort of the mechanism, but just by try and error and identify this toxin will just sort of paralyze the prey. But now through our understanding, actually we can really illustrate how it could work, because it prevents the neuromuscular junction synaptic transmission, even though the muscle, the transmitter acetylcholine can still be released, but the ion channel at the postsynaptic muscle no longer able to respond or to contract or to move. So you will just be paralyzed. Okay, and I will also mention a little bit later that how this toxin could work at a different aspect. But this is really an interesting way, how understanding the fundamental research can help us understand how the system could work. And in fact, in a few other cases, some toxins that can also provide tools help us to understand the fundamental biology of science. Okay, so initially you just by accident or by screen to identify some toxin, but just by understanding its target, then you recognize, you actually is the specific machinery inside the cell that's working in this way. And again, the most recent studies, okay, through structure studies by EN, by crystal structure studies, now we have a much better understanding at the atomic resolution how this acetylcholine receptor at the postsynaptic membrane recognize the acetylcholine. For example, we can really visualize this receptor, this pentamer, okay, this single amino acid or atom resolution, and also can identify the acetylcholine binding site by looking at the structure at the 2 different states. There is once the acetylcholine opens it, and one of the structure that is closed, we can actually picture how this ligand that working extracellularly can move the sort of pore inside to regulate the ionic flux. Okay, and that is again can also provide deeper understanding of this process. And because of, for example, we really know the binding site, we can also design better toxins or therapeutic agents to inhibit this ion channel or activate this ion channel by matching some chemicals interaction with these receptors. In fact, acetylcholine receptors is just one of the receptors that I illustrate. Again, it recognizes acetylcholine. But we have more transmitters, and they are recognized by different ion channels. For example, glutamate, that in the middle here, showing cartoon here, that again, glutamate is the major excitatory synapse transmitter in our brain, and it actually interestingly from the tetramers, different from the acetylcholine receptors, the ion channel, the nicotinic-like receptor, it has the pentamers. The glutamate forms tetramers, and also ATP in some specialized neurons or cells can also serve as transmitter. It will activate the so-called P2X receptor, they form trimers. And GABA acting on GABA A receptor, again, different GABA A receptor, different pentamer structures, but recognize GABA A, likewise the glycines. So the above part I mentioned mainly the postsynaptic receptors that, for example, how it can sort of being activated by acetylcholine. And that illustrates one of the general principles. Glutamate can activate this the corresponding glutamate receptor, but all those receptors are belonging to this so-called ionotropic, meaning that the receptor itself has the recognition site, binding site for acetylcholine, but at the same time, it's an ion channel. This acetylcholine binding will lead to this receptor to open up to allow the ion flux going through this port. But there's another major type of different receptor with the 7 transmembrane topology. That is, it has a 7 transmembrane helical going through the membrane, and it's called a G protein couple receptor, or also it's called a metabotropic receptor, because of the ligand here sort of a lot of times are the metabolites. And this receptor itself is not ion channel, but rather is binding to the ligand binding will also induce conformational change, and these conformational changes will recruit G protein, okay, and other intracellular signal pathway indirectly regulate ion channel as well as other protein machinery for gene regulation, for example. And now that we are going to discuss this second class of ion receptor, the metabotropic receptors. And so this is the classification of these two major receptors. On the left are the ionotropic ones. On the right are the metabotropic ones. And again, the same small molecule like acetylcholine or glutamate can activate multiple different receptors. And it's this sort of a diversity of the sort of receptors can create the diversity of response. Okay, and if you think about it, the nervous system really need to fine-tune to the response with different temporal response and with different magnitude. Need to be sometimes positive or negative, and they also integrate. And it's this diversity of the receptor at least contribute to part of this sort of a diversity of the repertoire of this response. And let me just back up a little bit. In fact, because the glutamate is the most important excitatory transmitter in our brain, let me talk about a little bit more about the ionotropic sort of receptors. So at a more later part, I will talk about the metabotropic. So the ionotropic receptors for glutamate in our brain can mainly classify into two categories. One is called AMPA, the other one is called NMDA. Okay, again, they are all the ionotropic glutamate receptors, all response to glutamate. have slightly different properties, and their naming are based on their agonist. That is, the AMPA receptor can be artificially activated by the AMPA chemical, and NMDA can be activated by the NMDA chemical. And they have different properties, and in fact, the NMDA is known to be a very critical glutamate receptor important for learning and memory. Okay, and similar to acetylcholine nicotinic ionotropic receptors, the glutamate ionotropic receptor activation, it will also allow the positive ion to go through, and therefore it's an excitatory one, even though structurally it's not a pentamer, it's a tetramer. And the AMPA and NMD receptors actually working together to mediate this excitatory transmission of glutamate in our brain. And they have a slightly different property, even though they share the similar sort of a structure and also similar sort of a positive ion conductivity. And one of the major difference between AMPA and NMD receptor is that NMD receptor is more sophisticated. It's not only being regulated by glutamate, but also at the same time regulated by membrane potential. In the resting membrane potential condition, even when the sort of glutamate being applied, NMD receptor will not be activated. Okay, glutamate AMPA receptor will be activated, will allow positive ion to go through, but NMD receptor will not. And it turns out, because of this some slightly different structure than the AMPA receptor, the NMD receptor in the resting membrane potential, in the presence of glutamate, there's still a positive ion mechanism in this ion permeation pathway will prevent this ion channel from conducting. Only during the postsynaptic membrane to be in a depolarized condition, then the mechanism will be sort of removed. Because if you think about it, that the membrane potential inside is very positive, the positive charge will sort of somehow repel the positive charge sort of mechanism. Then this ion permeation pathway will sort of conduct, and it will allow the positive ion going through. And also another different part of this NMD receptor is that it has a higher permeability to calcium than the AMPA receptor. So you will allow a lot of calcium coming in. So then you ask, this seems to be very complicated. Why nature evolved this process, and what can it do? One of its unique function of this NMD receptor, because of its sort of a special property, is serving as a coincidence detector. If you look at the cartoon on the left here, again, for the NMD receptor to be activated, it essentially requires two conditions. It requires the presynaptic neuron to release glutamate. It also requires this postsynaptic neuron to be activated first, depolarized. Therefore, in the postsynaptic membrane, the NMD receptor in that cell can serve as a coincidence detector. It integrates two different inputs. Okay, it requires a presynaptic input of glutamate release. It requires another input that this postsynaptic neuron, for example, through other mechanism to be activated. Then this NMD receptor can activate it and then allow calcium come into the cell. And this calcium, again, is a very important second messenger, can lead to the biochemical pathway to turn on, for example, by activating some enzyme like kinase or sort of phosphatase, and therefore you can create memory for the cell. In fact, we are not going through in our lecture, the NMD receptor knockout experiment by removing or blocking NMD receptor in some special hippocampal cells, neurons. Actually, we can demonstrate that it can lead to some knocking out mice do not have a good memory. Okay, so the NMD receptor is especially important for sort of create memory to detect different input, for example, the conditional inputs. Okay, and this graph showing here, that's how people really characterize the special property of NMD receptor with and without magnesium. For example, in the red is with the magnesium. At the negative membrane potential, these NMD receptors are blocked. But only when the magnesium is removed, that the NMD receptor is conducting. Okay. Similar to the presynaptic sort of scaffolding protein that I mentioned, that this ring protein, the postsynaptic membrane also has this scaffolding protein, especially corresponding to previously I mentioned the electrodense material. Okay, so here showing that in a cartoon fashion, that you have the AMPA receptor, MD receptor, and there's this PSD95 at the postsynaptic site, and also with a lot of other scaffolding protein and connect with the actin cytoskeleton as well as the cap kinase 2. It really creates the sort of essential sort of protein machinery also close to the postsynaptic membrane to mediate, for example, the signal transduction. And also part of the essential ones is the cap kinase 2 here, again, can be activated by calcium. Okay, so that is the fundamental understanding so far that the people have been reached for our central glutaminergic ionotropic receptors. And now let me just spend some efforts talk a little bit about the inhibitory receptors. Again, the GABA is the sort of the major inhibitory transmitter. It can also, just like a glutamate, can also activate ionotropic receptors as well as the metabotropic GPCRs. But for the ionotropic receptors, the GABA will activate the receptor. The receptor is unique in a way, even though it also has a pentamer, but it will allow different ions, the chloride ions, to go in through it. And chloride is negative charges, okay? And in a normal condition, the chloride concentration is relatively high outside of the cell. So once this GABA receptors being activated, chloride will come into the cell, bring the negative charge, and will make the membrane potential sort of not easy going to depolarize to reach the threshold. So activate the GABA receptor by GABA will maintain or will prevent the neuron from firing action potential, and therefore is a sort of a inhibitory transmitter. And this is showing one of the early experiments that done in 1950s, that people initially first identified this inhibitory sort of spinal cord neuron that will have this inhibitory GABA response. Okay, so now I covered almost all of the major ionotropic receptors that can be activated by neurotransmitters, and there is acetylcholine and also glutamate and GABA. Now I switch gears to these GPCRs, and GPCR again is a 7 transmembrane proteins. In our human genome, there's close to 1,000 GPCR. Most of them, more than close to 500 of them, are in our nose, in our sort of neuroepithelial cell in the nose that are sensing the odors or odorants. Okay, important for our smell. It turns out animals like mice has even more GPCR. For example, in mouse nose has more GPCR than human. Okay. In addition to this sort of GPCR to sense odorant or sense taste, there will be lectures about that. There's also this GPCR that mediates our synaptic transmission response. And the most recent studies, again, we have already achieved at the atomic resolution how this GPCR structure sort of forms and how it actually forms the downstream G protein complex. And for example, Branco-Bilka, this is at the bottom. Most recently, actually got a Nobel Prize by playing a pioneer role in solving the GPCR structure, namely the beta-2 adrenergic receptors. And it all has this typical 7 transmembrane topology with this N-terminal facing the extracellular part and C-terminal facing the intracellular part. And on the figure B, actually, I think that is one of the major contribution of Brian, where through years' effort, he crystallized the G protein, and in this case, is the beta-2 receptor with the G protein. And the G protein intracellularly is a trimer. And in sort of always golden-like structure, illustrates that is the sort of G alpha protein. And at the bottom, sort of bluish ones are the 2 sort of beta and gamma subunit. And this is really interesting. You have the ligand that will recognize binds to the GPCR, the G protein coupled receptor, extracellularly, like acetylcholine, like glutamate. And this GPCR, the G protein coupled receptor, will rearrange its structure and will engage the intracellular G protein. And therefore, that is how this signal transduction really occur across the membrane. That is how the extracellular signal changes can lead to the intracellular chemical reaction. In fact, the GPCR is the major drug target for the world. There are more than 30 drugs that are all target of GPCR. Again, this is a part of the pathway that you can sort of understand and use for therapeutic purpose. You use the sort of the drugs to control the GPCR and then change the intracellular signals. And what is being unique about this GPCR signal is that acetylcholine and glutamate, once it binds to the GPCR, it will engage of this G protein, and the G protein, the G alpha, is sort of a GTPase, and initially it has this GDP bound inside. Okay, and GPCR activation will make this G alpha protein change the conformation and will exchange the GDP with GTP. Okay, and the G alpha protein will have a different conformation, and different conformation will allow you to engage with other important effector proteins inside the cell. For example, some cyclase, the cyclic AMP synthesis, to generate cyclic AMP. And this is illustrating some of these known intracellular signal cascade. That is the activate G protein. Again, the G protein has different isoforms. The depends on the receptors, depends on the GPCRs, and depends on how the cell expresses the proteins. There's this excitatory or stimulus G protein called GS. And why is it called GS? S means stimulus, stimulatory, and that will activate the AC, the adenyl cyclase, to sort of catalyze ATP into cyclic AMP. Okay, there's also the GI GS protein. Okay, again, depends on different GPCR. The same acetylcholine or glutamate depends on different GPCR that it can either activate GS or activate GI. And therefore, this acetylcholine or glutamate in this case is called modulator because it can either make the second AMP level high to make the cell usually more excitable or inhibit the second AMP. Okay, and the second AMP is this small molecule, then can further activate kinase. In this case, is the PKA, protein kinase A. And the protein kinase A initially is inhibited by this regulatory domain, okay, R here, and the C is the catalytic domain, okay. The kinase will add a phosphate to modify a protein, okay. And once the second AMP binds to it, the second AMP will bind to this regulatory domain, PKA. Again, will make conformational change of the regulatory domain. And the regulatory domain initially is inhibiting the catalytic sort of enzyme, and now it's free. And this enzyme is a kinase. The kinase, the definition is to transfer a phosphate to a critical site in its substrate that will lead to the phosphorylation of its target. And in this case, for example, a voltage-gated calcium channel. And this phosphorylation will also make changes of this ion channel, this voltage-gated calcium channel. And that will, for example, activate or inhibit this channel to conduct calcium. And therefore, the calcium will be regulated, will lead to, for example, if it's in the heart muscle cell, you will change the muscle sort of contraction, okay? And in fact, beta-arginine receptor, the beta blockers, is probably one of the most widely used drugs to treat the cardiovascular diseases. Okay, and it's again tap into the known sort of synaptic transmission, because the heart, just like many sort of organ in the body, is also controlled by the neuron through the synaptic transmission. The neurons, specifically the autonomic sort of nervous systems that control the bodies, and norepinephrine is being released into the muscle cell, including the heart muscle cells, through the neurons. But the beta blockers can work in a way to prevent the action of norepinephrine, and in a way to control the muscle cell, and in a lot of cases to allow more calcium influx, and then to have a more beating or more stronger contraction of the heart muscle cell. And in addition to the G protein that I mentioned, there's also GQ, another G protein. And again, through the similar but different downstream target enzymes, this GQ protein has a different conformation that it will, once it's been activated through this GTPase, the G-alpha GTPase, it will activate the phospholipase C, yet another enzyme. And this phospholipase C will cleave the PIP2, this membrane lipid at the plasma membrane, and release IP3. And this IP3 can diffuse and activate the intracellular organelles, the ER, the endoplasmic reticulum receptor, and allowed this intracellular calcium store to release calcium. And then calcium can also work on the enzymes. So therefore, you can see that it's critical to understand how the extracellular signal can get converted into the intracellular signal, because the cells are shielded by this plasma membrane, usually are not permeable to a lot of chemicals. And the ion channel is one of the special types that I mentioned earlier, that the acetylcholine glutamate binding will allow the positive ion or negative ion to go through to change the membrane potential. But the metabotropic GPCR is a non-conducting receptors, but through this conformational change, you will engage of diverse G protein alpha here, I mentioned GS, GI, GQ that will allow the signal cascade. Okay, and in fact, the G beta gamma itself can also serve as a regulatory for some ion channel. Okay, for example, it's known that the G beta gamma through this primer G protein. The G protein again has the alpha subunit and beta gamma subunit, and beta gamma subunit can also interact with some ion channel to control, for example, potassium channel, calcium channel, and sodium channel. And therefore, this transmitter that I mentioned, there's a lot of them, and its receptor has also a lot of them, and intracellular signaling, there's also a lot of them, and it's really this computer road that it can generate countless combinations, and it really depends on the specialized cell, and especially the expression of a certain gene that the nervous system can create unlimited sort of response, either being excitatory, inhibitory, and can control the cell response. And also this GPCR, not only the metabotropic receptor I mentioned at the postsynaptic site, okay, it can sense the acetylcholine, but also there's this metabotropic receptors, GPCR, at the presynaptic site, and it can also sense the transmitter release either from other neuron or from itself. And again, through the signal transduction, it can control the transmitter release by itself. For example, as I mentioned, the acetylcholine, the GABA receptor, the GABA A is the ion channel, okay, but the GABA B receptor is the GPCR. Once the GABA being released from other neurons, for example, from an adjacent GABAergic neuron, that it can activate the GABA B receptor, and the GABA B receptor either can, turn on the potassium channel to allow the membrane potential to be hyperpolarized, positive ion going out, okay, membrane potential will go down, okay, or control the calcium channel to allow less calcium influx. Then this neuron will be relatively silent, and there's less calcium to trigger transmitter release. Then this neuron will not release or release less transmitters, so it will be more inhibitory effect. And there's also other GPCR, for example, serotonin receptor, through some pathway can will inhibit the calcium, potassium channel, will make this cell more excitatory. And these GPCR are indeed located both at the presynaptic membrane and postsynaptic membrane. And once the transmitter release, it really depends on the combination of those transmitter and the ion channel. The neuronal response can either be excitatory or inhibitory. And that actually is one of the challenges related to my own research. Our own group actually are making indicators that can allow us to look at the response of acetylcholine, dopamine, and once those transmitters being released, the neuron will light up, and that will already help us to understand what transmitter being released. And combined with other tools, we can have better understanding what is the effect of this transmitter being released. Okay, so just to summarize a little bit of this GPCR signaling, okay, the GPCR different from the ionotropic receptor itself do not allow ion to go in through the membrane, but through engaging of G protein, and G protein, the different kind, and G protein has a diverse substrate or target enzymes that it can regulate a variety of processes. And so why nature evolved this so complicated systems? Well, if you look at the figure on the right, if you have the ionotropic receptor, because the protein itself is an ion channel, so it actually can make the reaction happen really fast. Okay, so the ion channel being regulated by acetylcholine glutamate, immediately conformation change, it allows the ion going through, so that can achieve fast speed, muscle contraction, fast computation. So that's good. But also in the nervous system, there's also a lot of reactions are relatively slower, and therefore nature actually evolved these systems to have through the mediate metabotropic receptor pathway that can either be more positively regulated or negatively regulated. And also through this process, you have a small second EMP or sort of other second messenger as a way to amplify the signal. So the response either can be slower, but also can have a larger response. And this altogether can shape the nervous system's response. In fact, the calcium influx into the cell not only can change the kinase and modify ion channel, but can also lead to the gene expression changes. And that is sort of one of the mechanisms that is being believed how the long-term memory is really stored. Right? You have everyday reactions, but how you, after 20 years, you still remember. Actually, the nervous system releases transmitters through synapse, but this modification of the synapse through this neuronal communication in certain conditions will, through the cascade, illustrated here, for example, activates the kinase, and the kinase will integrate the signal, eventually activate the transcription factors, for example, the phosphorylation crab. And this transcription factor will turn on gene expression, and that can last for very long term changes of the cell's gene expression, and that can be long lasting, and the gene will lead to the protein being translated eventually, and that can lead to the change of the neuronal network, and that is believed to be the mechanism of the long-term memory. Okay, so my talk has been quite intense. In the last 5 to 10 minutes, I will finish the lecture again. The detail of this synaptic transmission can be a course for 1 semester, and it's really highly being compressed in this way, and it's challenging to balance the amount of materials and exact experiments to sort of that have lead to these discoveries, to balance all these materials together. But the nervous system, for a neuron's perspective, neuron actually has this complicated shape. It's a polarized cell, which is mentioned in the first lecture. It actually has this sort of dendrites and long axons to send out information, and the dendrites to integrate information. And indeed, at the synapse, at a different part of the neurons are really integrated to process the information. Okay, for example, this is sort of a single neurons that express a GIP, and if you look at this neuron, you can see this spine, that this protrusion. Okay, but the neuron actually is even more complicated, and this is showing on the left a typical pyramidal cell. Okay, and in our human brain, we have a cortical region. The pyramidal cell that might span 6 layers. Okay, it has its neuronal cell body reside in one of layer, for example, layer 5. Okay, and it also has this, apical dendrite and basal dendrite. And in fact, in the nervous system, the synapses are arranged in a very sort of sophisticated ways. It's not really random. Okay, and as you can see here, there's different presynaptic neurons, and here labeled are mainly the inhibitory neurons, and they have different shape, and importantly, they also have different property, and they innervate different part of the dendrites. Like on the top, this Martinotti cells that they usually innervate more sort of top layers. And also this basket cell, the name why it's called basket is because of the neurons will usually innervate the cell body, and the cell body is this sort of like a spherical like shape. It only sort of highly innervate the cell body. It forms like a basket like structure, so it's called basket cell. So you only send the synapse there. And if you check mice, different mice, and using this molecular marker to stand it, they are just all the same across different. And this information are strategically processed at a different synapse. And also this is really intriguing chandelier cell, okay, showing here on the right with one of the chandelier cell labeled by GAP. And this is really a unique chandelier cell, and in a way that it has this axonal synapse just in a way the postsynaptic neurons axon initial segment, okay, where the action potential is generated, okay, and on the right, as you can see here, that this chandelier cell actually has many presynaptic axons, and each axon, in a way, one postsynaptic axon initial segment, and in a way, it's believed that this release of the transmitter there is GABA that will inhibit postsynaptic neuron to generate action potential. And this really chandelier cell structurally similar like chandelier, right? And it's believed could serve as a sort of vital power, because a single chandelier cell, once being activated, it will send like 500 or 300 vital power to the axon initial segment, to the postsynaptic excitatory glutamate neurons, and this axon, the pyramidal cell cannot fire. Okay, and exactly what is the function of the chandelier cell in our conscious, in our brain, is still unknown. But this really illustrates that this diverse cell type through this conserved molecular mechanism that I mentioned, that is the transmitter release, either activate or inhibit the neurons, that can really control the information flow. And finally, why do we care? Why do we care about the SNARE transmission? And I mentioned there's this SNARE protein, Synaptotagmin, and there's this layer arranging and diversity of neuronal types. Why bother? Why do we what is the benefit of knowing that? Well, one of the things I think is this is just for fun, curiosity. Okay, knowing how, knowing human's brain, and we use our brain for our reasoning and understanding the world, and knowing how this neuronal cell communicate. I think it's just fascinating by itself. But let me just give you one example of its practical application, and that is the neurotoxins. Okay, again, I mentioned a little bit earlier about the curare, plant-derived chemicals that can, being used, by people in Central Americas, many years ago. But in fact, nature evolved many toxins. Okay, spider, scorpion, snake, a pufferfish has also those toxins. And this toxin, in a way, is fascinating how they work. And also, by knowing the mechanism, some of them actually can be used, for, human purpose. For example, the alpha bangrol toxin that from snake on the left, that actually is known to be important to block the nicotinic receptor in the neuromuscular junction, and that is how the snake, they injected the venens, and then that can paralyze the prey. Okay, and in fact, this PowerPoint illustrates many of those neurotoxins that can work on different targets, starting from the ion channels, like what you guys have learned from action potential generation. There's the sodium channels being targeted by the tetraneurotoxin. Okay, that again block the voltage-gated sodium channel, and therefore there will be no action potential being generated. Okay, and therefore this is useful for the animal to capture their prey. Okay, at the block the generation of action potentials. Okay, and of course, this can help us to understand the role of voltage-gated calcium channel, sodium channel. It turns out in humans there are certain pain sensing neurons. They have specialized voltage-gated sodium channels. Okay, and therefore design toxins just target those pain sensing sodium channels without altering other sodium channels will be a useful approach to design drugs for anesthesia or to reduce pain. Okay, and again, this is an active research area. Okay, there's also on the left here, there's also conotoxin from snails, and they are known, some of them sedatively block voltage-gated calcium channel. And because calcium influx, now we know, is important for transmitter release, and prevent of these voltage-gated calcium channels can also prevent transmitter release. No calcium triggering, no transmitter release. So similarly to alpha-beta toxin will not prevent transmitter release, but transmitter release cannot activate the postsynaptic neuron. Now this conotoxin can prevent transmitter release, and it can also prevent synaptic transmission of occurring, no muscle contraction, no escape. Okay, and it's useful for snail, actually some sea snail to have. But it turns out some of these snail toxin and their derivative are already FDA approved drugs for sort of curing headaches or migraine. It turns out there's some specialized voltage-gated calcium channel are involved in transmitter release, and not all the voltage-gated calcium channel are the same, just like the sodium channel I mentioned. And it turns out that the fish use those voltage-gated calcium channel that in their muscle for their muscle contraction, but in human, those conotoxin sensitive voltage-gated calcium channel in the neurons related to headache, migraines. Therefore, even though conotoxin is deadly for fish, but for humans can be used to silence those headache-related neurons and can eliminate headache or migraine, and being used for intractable headaches for human. Okay, and there are also one last thing I want to mention, like the Botox on the right. Okay, again, the Botox toxin are the toxin that will selectively cleave the SNARE protein, synaptotagmin syntaxin I mentioned. Okay, and once they've been cleaved, there will be no transmitter being released. And it turns out a very brilliant usage for it is in the for human, or especially mostly women, to eliminate wrinkles. It turns out that the wrinkles are generated again by their muscles, and which are innervated by nerves. Okay, and once you poison the neuron that release transmitter to sort of to loosen the muscles, the wrinkle is gone. Okay, so this Botox has a huge business in human society, and the mechanism is it will get into the neuronal nerve terminal, and it's a protease that will selectively cleave SNARE protein and prevent transmitter release, and therefore there's less transmitter release to excite the muscle, and then there's muscle contraction and loosen, and that will reduce the wrinkles. Okay, so of course, now knowing the mechanism, and you can appreciate the Botox application needs to be very, very careful. You only need to use the dosage in the part of the wrinkle part that you want. And if we spread to other muscles, for example, the muscle control your movement, the muscle control your breathing, then you are going to be a dead man, okay, or dead woman, okay. So knowing the molecular mechanism of the synaptic transmission actually can help people to design strategies for therapeutic purpose. Finally, I want to summarize my today's talk in the last 2 minutes. Through this almost close to 2 hours talk, I going through to the neurotransmission, and the mechanism of the synaptic transmission at the presynaptic side can be summarized by that the action potential will propagate and arrives at the presynaptic terminal, and that will trigger transmitter release. And the transmitters are packed in a discrete packets in the synaptic vesicles, and the release really requires the transmitter, the synaptic vesicle fused with the plasma membrane, presynaptic plasma membrane, and this process is controlled or triggered by the presynaptic calcium entry, and the calcium entry requires the presynaptic voltage-gated calcium channel to sense the sort of depolarization generated by the action potentials. And also, inside the presynaptic terminal, there are specialized proteins, SNARE protein I mentioned, for example, Botox targets it, right? And SM proteins that locate either in the presynaptic membrane or in the synaptic vesicle, just right at the place, and then sort of a mediate vesicle fusion. And Synaptotagmin is serving as the calcium sensor to trigger the vesicle fusion. And the presynaptic terminal is not a random structure, but rather there's some additional scaffolding protein that organize in a highly structured domain called the active zone, where it's highly localized the voltage-gated calcium channel to the synaptic vesicle, and also with all this fusion machinery right there. And nervous system uses many transmitter, and different transmitter are in different neurons, and important ones are the glutamate excitatory transmitter in our brain, and acetylcholine excitatory transmitters in our motor neuron, and GABA at the sort of inhibitory transmitters. And neurotransmitters are efficiently cleaved from the synaptic cleft by either enzymatic cleavage, that is for the acetylcholine, or transport. And also glial cell also participate in this process by sort of reacting. And in the postsynaptic side, the postsynaptic receptor to sense the transmitters can be classified into ionotropic or metabotropic. Okay, and the skeleton acetylcholine receptors is the nicotine-like, is a ligand-gated ion channel with the pentamers, as I mentioned. And AMPA and NMDA receptor are the glutamate receptor in our central nervous system. They are ionotropic ones. And NMDA, I especially emphasize, might be important for learning and memory because of its unique sort of nature, the detector. And metabotropic receptors are all G-protein coupled receptors, and with this unique 7 transmembrane topology sensing different transmitters. And its downstream G-protein is a trimer, can be classified into the alpha and beta gamma, and they are all membrane associated. Okay, so they are close at the membrane and can trigger diversified downstream target pathways, and then if the downstream target pathway can include ion channels, you can also change the membrane excitability. But there's also additional pathway, including gene expression, that can be more long-lasting. And also metabotropic receptors can also locate in the presynaptic terminals to control transmitter release. And I mentioned the last part, the postsynaptic depolarization can also induce gene expression. And again, it's related to long-term memory. Okay, with that, I will end with my lecture. I think the TAs will help with the answering the question, and I believe I also sent my prepared PowerPoints TA, and that can be distributed. Okay.
Yeah, thank you so much for giving that lecture. Are you able to stick around for a few minutes to answer some of the students' questions?
Yes, okay.
Okay, that sounds great. So like we've been doing, if you guys have any questions, you can just raise your hand and I'll call on you.
Or you can mention their question to me and I can answer. Any questions?
I guess maybe there are no questions.
Okay, all right. Well, I think if they have questions, and you guys can help answer. Okay. All right. It's a pretty intense talk again. And thank you all for attending. And I hope that will be useful for you guys. Yeah.
Yeah, thank you so much, Professor.